Imagine finding creatures so strange, they almost seem to come from a fever dream. They were real organisms. They lived in Earth's oceans roughly 600 million years ago, during the Ediacaran Period. If we could descend into those waters, there would be no fish, no crabs, and no coral reefs crowded with movement. Across the seafloor lived organisms so unfamiliar that scientists still debate what many of them actually were. Oxygen levels in the oceans and atmosphere were still relatively low compared with later eras. Much of the seafloor was covered by microbial mats, living carpets of bacteria and other microorganisms that stabilized the sediment and created a surface unlike most modern seabeds. Across those mats, something extraordinary was beginning to happen. For the first time in Earth's history, large multicellular organisms were becoming widespread and visible on the seafloor. Some rose above the sediment, some lay flat against it. Others moved slowly across its surface, leaving traces behind. Their bodies experimented with forms that do not fit easily into the categories we use for modern life. Some may have been animals. Others may have belonged to extinct branches of multicellular life. In many cases, scientists still cannot agree on exactly what they were. This is what makes the Ediacaran so strange. It was not simply a world before familiar animals. It was a world where life was already complex, but organized according to rules that often feel almost unreachable from the present. Among the most iconic of all Ediacaran fossils is Dickinsonia. Its body resembled a flattened oval quilt, divided into repeated units that gave it a strange, ribbed appearance. For decades, Dickinsonia was often portrayed as one of the earliest animals, and sometimes even as a possible bilaterian, a distant relative of the lineage that would eventually include worms, insects, dinosaurs, and humans. The evidence has never been simple. Some studies of fossilized organic molecules found compounds interpreted as cholesterol derivatives, supporting an animal affinity. Other analyses have argued that Dickinsonia and its relatives, the enigmatic proarticulates, may not fit comfortably within any known animal group. A few researchers have even suggested comparisons with certain fungal lineages. So Dickinsonia remains one of the great problems of Ediacaran paleontology. Charnia opens a different kind of mystery. This frond-shaped organism lived anchored to the seafloor and ranks among the oldest large multicellular organisms ever discovered. It belonged to the rangermorphs, a group defined by branching bodies built in repeated patterns. Each branch was composed of smaller branches, repeating the same design at multiple scales. The result was unlike a leaf, unlike a coral, and unlike any ordinary animal body. Rangermorphs dominated many Ediacaran ecosystems. They formed tall, delicate structures above the seabed, increasing surface area in a world where absorbing nutrients directly from the surrounding water may have been an important strategy. Yet their relationships to modern life remain deeply uncertain. Fractofusus belonged to that same strange world, but lived in a different way. Instead of rising upright like Charnia, Fractofusus lay flat against the seafloor, resembling a leaf pressed into the sediment. Fossil assemblages show that it often reproduced clonally, forming clusters of genetically identical descendants across the seabed. Other Ediacaran organisms seem to approach familiar animal shapes. Spriggina is one of the best examples. Its elongated body appears divided into repeated segments, and its enlarged front region almost looks like a head. Because of this, early researchers often compared it to ancient arthropods. But appearances can mislead. Spriggina belonged to the Proarticulata, a group whose body organization differs from that of true bilaterian animals. What look like symmetrical segments are arranged in a peculiar offset pattern, unlike the clear left-right organization seen in modern arthropods. A similar problem surrounds Parvancorina. This small organism had a shield-shaped or anchor-like body. At first glance, its front region can look surprisingly similar to the head shield of certain early arthropods and trilobites. But the resemblance stops. There is no clear sign of legs, articulated segments, or the defining features of true arthropods. Some researchers have suggested that Parvancorina may have oriented itself with ocean currents. Others have questioned how much it moved at all. Yorgia adds another layer to that problem. This proarticulate could reach around 25 centimeters in length, making it large by Ediacaran standards. Fossils show that it moved across microbial mats and left distinctive feeding traces behind. At first glance, Yorgia looks like an animal with a head and a body axis. What appears to be a head may not have functioned as one at all. Yorgia is a warning against reading these fossils too quickly through modern anatomy. Not every organism from this world feels equally distant. Kimberella appears more familiar than many of its neighbours. Its body has often been compared to that of a slug or limpet, and fossil traces suggest that it scraped microbial mats while feeding. Many paleontologists consider Kimberella one of the strongest candidates for an early member of the broader mollusk lineage. If that interpretation is correct, it may represent a distant precursor to the evolutionary branch that would later produce snails, squid, and octopuses. That possibility makes Kimberella important for a different reason. It suggests that some lineages leading toward recognizable animal groups may already have been present before the Cambrian Explosion. Ikaria is much smaller, but possibly even more significant. This simple worm-like organism was only a few millimeters long. Fossils and associated burrows suggest an active animal capable of moving through sediment. It had a front end, a back end, and bilateral symmetry. A body plan that would become one of the most important patterns in animal evolution. If Ikaria was indeed among the earliest known bilaterians, then its importance is enormous despite its tiny size. Much of later animal diversity depends on the basic organizational principle represented by animals like this. The Ediacaran was not only producing strange forms, it was also producing some of the basic instructions that later animal life would build upon. Hautia preserves another possible early innovation. This organism, probably related to cnidarians such as jellyfish and sea anemones, contains a complex network of fibrous structures interpreted as muscle-like tissues. If that interpretation is correct, Hautia may preserve some of the oldest evidence for this kind of organization in the animal kingdom. Arkarua points in a different direction. This tiny disc-shaped organism displays five-fold symmetry, a feature that has led some scientists to suggest a possible relationship with the ancestors of echinoderms, the group that today includes sea stars and sea urchins. The interpretation remains debated, but the possibility is important. Tribrachidium shows just how experimental symmetry had become. Its body was organized around 3 spiral arms arranged in rotational symmetry. No modern animal has a body plan quite like it. Its unusual form has led some researchers to compare it with ancient relatives of echinoderms, although its exact relationships remain uncertain. What matters is that the Ediacaran seafloor was already testing body architectures that would not become common in later animal history. Andromeda belonged to that same broader pattern of experimentation. Named after the spiral structure of the Andromeda galaxy, it possessed 8 curved arms radiating from a central region. Other organisms explored life fixed to the seafloor. Ikaria was a globular organism anchored to the substrate, probably functioning as a simple suspension feeder, somewhat reminiscent of a polyp. Thectardis had a cone-shaped body fixed to the seabed. Ausia presents a different possibility. Its body consisted of repeated chambers arranged in an orderly pattern, possibly forming an internal filter system. Some paleontologists have proposed that it may have been related to early tunicates, the group that includes modern sea squirts, and belongs near the broader lineage leading to vertebrates. Namacalathus marks another major shift. This organism possessed a goblet-shaped skeleton perforated by openings. It ranks among the earliest known organisms to produce a mineralized skeleton, a feature that would become increasingly important during the Cambrian. Its anatomy is strange and difficult to place, but many researchers have suggested affinities within the Lophotrochozoans, the broad animal group that includes mollusks, annelid worms, and brachiopods. Phoenicia preserves evidence for another important development. This tubular organism lived in dense clusters across the seafloor. Studies have suggested that these aggregations may represent one of the oldest known examples of sexual reproduction in multicellular organisms. Other fossils remind us how unstable Ediacaran classifications can be. Philozoon was originally placed among the proarticulates because of its flattened, segmented appearance. More recent analyses have suggested possible closer affinities with rangeomorph organisms such as Charnia. Onega remains even more isolated. This discoidal organism possessed lobed structures on its body, but its biological identity is still uncertain. Bomachelia is another difficult fossil. Attached to the substrate by a thick stalk, it had an oval body with concentric internal structures and lateral frond-like appendages. Some studies have suggested affinities with rangeomorph organisms, but its biology remains poorly understood. In fossils like Onega and Bomachelia, the Ediacaran becomes especially frustrating. The forms are preserved, sometimes in remarkable detail, but their deeper biological meaning remains hard to reach. Ivesheadia takes that uncertainty even further. At first glance, it appears to be a distinct organism, a flattened circular disc, almost like a collapsed body pressed into stone. Some specimens have even been compared to a strange prehistoric pizza, and the comparison is surprisingly accurate. For a long time, fossils like this were treated as members of the Ediacaran biota. But many researchers now suspect that Ivesheadia may not represent a species at all. Instead, it could be a taphonomic artifact created by the decomposition and collapse of other Ediacaran organisms after death. Among the most visually striking organisms of the period was Primocandelabrum. This organism rose from the seafloor on a central stalk, with multiple branching arms extending upward. Its shape has often been compared to a candelabrum. It was a living structure anchored to the seabed, likely filtering tiny particles from the surrounding water. Placed among rangeomorph-like organisms, Primocandelabrum shows how far Ediacaran life could push branching architecture. Taken together, these organisms reveal a world far more complex than the phrase "before the Cambrian Explosion" often suggests. The Ediacaran was not an empty waiting room for modern animals. It already had movement, experiments in symmetry, early skeletons, and large communities spread across the seafloor. Some organisms may represent early members of lineages that still survive today. Others may belong to branches of life that vanished completely, leaving no living descendants. Many of their relationships remain unresolved. When the Cambrian Explosion began around 539 million years ago, animals with skeletons, limbs, complex eyes, predatory behaviors, and more recognizable body plans would rapidly diversify and transform Earth's ecosystems. But beneath that famous radiation lies an older and stranger chapter. The Ediacaran world was not simply primitive. It was complex in a way that no longer exists. And that is why its strangest question still remains open. What exactly were they?